Thanks, Niels, for the invitation. I'll be presenting today on behalf of the Vanderbilt Biomic team, and I'll be describing to you our multimodal imaging pipeline with a focus on how we initially developed the methodologies in 2D, as well as the accompanying software and tools that we use to both integrate and interrogate those assays, and then how we're actually dragging our approach into the 3D realm. And shown here is just the different assays that we use as part of our multimodal pipeline. And there's a brief description of each one at the bottom. And so a couple of things that I just want you to take away from this slide is that we're actually assaying a number of different analytes. So we can basically profile lipids, small metabolites, glycans, transcripts, proteins, as well as morphology markers. And within each of these assays, we're also dealing with sort of a large variation in terms of the molecular coverage that each assay can give us, as well as a really diverse spatial scale that we're sampling across. And this presents some tricky challenges for us as we're trying to do true data set integration and allowing for that mining of data from one modality to the next. And so if we sort of frame our pipeline in terms of the molecular coverage and spatial specificity, we see that those two are actually inversely related. And so we have some very robust high molecular coverage assays, including Visium, where we have whole transcriptome coverage, as well as imaging mass spectrometry, which is sort of our workhorse technology that we use in our lab. And here we can sample things such as lipids, metabolites, as well as glycans, small molecules. And for each of these, while they give us really robust molecular coverage, we're sort of constrained by the spatial resolution to about a 5 to 10 micron spatial resolution. We also have some more targeted methods, including Xenium and Codex for spatial transcriptomics, as well as spatial proteomics, where we can get to cellular or even subcellular resolution. But the trade-off there is that we're obviously not sampling nearly as many analytes, so the data set, the molecular coverage is not quite as robust. And then rounding out our pipeline, we have just some very standard, straightforward microscopy techniques, including histological stains so that we can, pathologically assess our samples. And then the last one is actually this autofluorescence microscopy technique. And this is actually incredibly key for two aspects of our pipeline. And the great thing about it is that autofluorescence microscopy is essentially a free technology. All you need is a slide and a slide scanner, and you can collect autofluorescence microscopy on every single section. And we'll see that, autofluorescence microscopy is actually essentially the common language of a data set in terms of you can collect it on every single tissue section in a stack. And then you can use that to essentially co-register as you go from section to section. And so that's the first key piece of this autofluorescence microscopy, is it allows us to do serial section co-registration. It also allows us to essentially co-register modalities that are collected on a single slide. And so what I'm showing here is sort of our most standard multimodal workflow. And this is one that we routinely collect on our samples. And so what we do is we collect that initial autofluorescence microscopy image. Then we can do an imaging mass spec experiment and get the molecular profiles. Then we'll collect a second autofluorescence microscopy image. So this is a post-AF image. And then we can overlay our histology stain on top of that. And the key to collecting that pre and post autofluorescence image is that they're essentially identical in terms of their coordinate space. But the pre-AF image has a lot more intense markers and sort of the crisper resolution, whereas the post-AF actually shows the ablation craters that were created as the laser is rastering across the tissue during that mass spectrometry experiment. And so we can leverage the fact that we have the exact location of those mass spec pixels and actually co-register it with the pre-autofluorescence microscopy. And now we can give spatial context to that imaging mass spec experiment, essentially give it microscopy resolution to interpret that data. The second, and again, this is all driven by that autofluorescence microscopy. The second key component about that autofluorescence microscopy image is that early on, when we were developing this pipeline, we actually were able to select markers to identify specific functional tissue units within the kidney nephron. And so we selected markers that would identify the glomeruli versus proximal tubule, distal tubule, and so on and so forth. And so we could collect a series of multiplexed immunofluorescence images and use them to essentially train a model that would recognize each of these functional tissue units on that autofluorescence image. And so now, instead of having to collect an MXIF image every time we do an experiment, we can actually just use that autofluorescence image to do the segmentation. And so this saves us quite a bit of time, money, and resources to actually segment our data. What I'm showing you here on the right is an IMS image, a very high resolution, high quality IMS image generated by Martin Dufresne in our lab. And each of the colors being projected here actually represent a single lipid, so a single M over Z, a single ion. And what becomes somewhat immediately clear is that there are specific associations between lipids and the FTUs. And so if we kind of leverage the segmentation mass that we can create on that autofluorescence image, we can use that to essentially mine the lipid data and start creating molecular profiles that are specific to a given functional tissue unit. And we can do this using a process known as Shapley additive explanation plots. And what you can see is we can actually generate these lipid profiles specific to a given FTU. So we're essentially molecular barcoding or creating molecular fingerprints for each of these individual functional tissue units. And so you can see that, if you sort of break them down, the glomeruli have a distinct molecular barcode compared to a proximal tubule, compared to a collecting duct, and so on and so forth. And what is interesting here is, as we're creating these sort of molecular biomarkers, is now we can drill down into that data and look at the specific lipids that are associated with glomeruli, the specific lipids that are associated with proximal tubules, and start asking fundamental biological questions, such as, why is that lipid there? Why is sphingomyelin 34-1 so highly associated with glomeruli? Is there a structure-function relationship that we can discover from looking at these specific lipid profiles? And all of this is done just using that sort of very standard autofluorescence, imaging mass spec, autofluorescence, histology stain, all on one slide. And so, as I mentioned early on, we have additional modalities. Oh, sorry about that, little pop-up. We have additional modalities that we also want to add, orthogonal modalities, including xenium and codex, that we want to add to this workflow, and we want to do that in such a manner that allows for that cross-modality data mining. And so this is some work that is just about finished now. It's being spearheaded by Angela Cruza in our lab, along with a group of graduate students, where they are essentially collecting that autofluorescence IMS histology and then serial sections, including codex and xenium, and co-registering those serial sections in a manner that does allow them to drive that cross-modality data mining. So now, not only do we have the lipid profiles, but we can essentially layer over that the cellular profiles that are being generated by codex, and then the transcriptional information being generated by xenium. And now we have true crosstalk across our modalities. We can look at cell types as well as cell states and how all of these things are sort of changing in tandem and try to answer some deeper biological questions. Now, as we were doing this, we sort of came across a conundrum, which is, how do we preserve spatial fidelity? Because if we're co-registering from section A to section B, those sections are, of course, 10 microns separate from each other. And so there's a little bit of wiggle in terms of, the cells that are in section A versus section B. And so what we endeavored to do was to create a multiplex workflow. Can we multiplex some of these orthogonal experiments on top of the AF IMS workflow that we already have on that single slide? And so for that, Ty Pham, who's a postdoc in our lab, and he's here with us today, developed a workflow where he could take the slide out of the mass spectrometer and take it immediately into a codex workflow. And now we have a highly multiplexed series of events that are being detected on that same tissue. And when Ty compared the IMS codex workflow to just codex alone, he saw that the images that were generated were entirely comparable. And so from there, he was able to essentially do some clustering and assign cell types project those back onto the tissue section, and now we have specific cell types on that tissue section that have been identified. And then working with our collaborators at TU Delft in the Netherlands, Lucas Migas was able to use this Elastics whole slide imaging co-registration workflow, again, relying on that autofluorescence image that's taken in series to essentially co-register the lipid data with the codex data. And this allows us to do some really unique analyses, because now what we can do is not only can we do segmentation and definition of the cells, but we can actually generate neighborhoods. And we can take those neighborhood masks, similar to what we did with the autofluorescence FTU segmentation mask. We can use those neighborhood masks to now mine the molecular data that's associated with those neighborhoods. And this is actually generating some really interesting observations that Ty is putting together in a manuscript that's going to be submitted in the near future. But that then brings up sort of the next question of, if we're describing cellular neighborhoods, but we're only sampling in the X and Y plane, are we truly describing cellular neighborhoods? Because the cell obviously has neighbors in all 3 dimensions. And so that's what's really driving us to the next level, which is, can we start to build 3D volumes from all these 2D assays that we've developed? And in order to do this, again, we rely heavily on our collaborators at TU Delft, so Lukas and Ralph van der Plas, and they've created this sort of pipeline for doing that, for getting that co-registration for those serial sections and projecting the 3D volumes that are associated with it. I think probably the key part of this whole pipeline is the fact that they have these sort of QC metrics where they can measure their co-registration error, and then they can iterate and go back and refine the pipeline and refine the pipeline. Because of course, if you have, an error in that first step, and now you're propagating it over all those dimensions, that's going to become more and more significant. And so now I'm just going to show you a couple of examples of some of the 3D data sets that we've collected and how we've co-registered them and projected the 3D volumes. So our first sort of pilot experiment was very small. We just chose 3 MXIF markers that would distinctly mark 3 functional tissue units within the nephron. And we collected the data across, I think, 64 serial sections. And essentially just challenge ourselves, can we collect that data, co-register it, and project it in a 3D volume? And what you're seeing here is the projections that come out of this data. And we were really, really pleased with this, because now you can see on the one on the left here in green are the glomeruli. And now we're not just thinking about how does a glomeruli interact with its neighbors to its left and right, but how is it truly interacting across that tissue space? After completion of this small pilot project, we then expanded our MXIF panel so that we could capture not only all the FTUs, but also vasculature, and we could start asking some, broader questions about these neighborhoods. And so this was done across, I believe, 58 serial sections, and then projected into these 3D volumes. And now you can really start to see that this is, as a biologist, as a person doing the interpretation, this really speaks to me in terms of, now I can ask, some very complex questions about how are the different components of the nephron communicating with each other? How is nephron A communicating with nephron B? How are any of these nephrons communicating with the stroma, the vasculature, the interstitial space, and really get at some of those complex biological questions? Now, of course, doing a 60-section MXIF or codex for every single sample would be just absurdly priced. And so, we want to lean back into some of the methods that we developed early on and leverage that autofluorescence microscopy. And so this was just a small proof of principle experiment showing that we can also collect the serial sections for autofluorescence and project those back into the 3D volume as well. Again, since we already developed the workflows for essentially co-registering the autofluorescence with the IMS, then we know that we can drag this into our 3D workflow. Again, really relying on those autofluorescence images to allow this to happen. And what we can then do is collect 3D IMS images shown here. And this is just really a brilliant, stunning example of looking at these lipids, not just in that 2-dimensional plane, but in 3-dimensional space, and going back and asking and answering some of those questions about, why is this lipid associated with this region, or why is this lipid associated with this cell type? Of course, we've been very kidney heavy, and so I just wanted to demonstrate that we are not restricted only to the kidney. We can also collect 3D IMS data sets for other tissues. Shown here is the optic nerve and the retina, and so this is just an eye sample. I believe that there were 64 serial sections here, and identified a number of different interesting lipids that are associated with different structures within the eye. We can also use this to assay disease tissues. So this is a staph-infected murine kidney. This is 100 serial sections that were collected and then co-registered and 3D volumes rendered. And what's really interesting is not only can we identify, certain cell type markers such as hemoglobin fragment, but we can start to see differences in the lipids and the way that they are, enriched in either the healthy tissue that's surrounding the area. So that here, this phosphatidylcholine 38-2, whereas others, this phosphatidylcholine plasminogen is actually really enriched in the abscess. And it's not just a single layer of the abscess, it seems to be present throughout the abscess, which is incredibly interesting because this abscess is just the equivalent of a giant ball of biological snot, and being able to actually profile lipids through that giant ball of biological snot is going to allow us to start understanding what's actually happening within that abscess. And of course, I wanted to leave you with one more kidney image, and this one here, where we've collected the 60 serial sections, done the autofluorescence microscopy, and we're tracking a single lipid in this case, in this projection, and that's that sphingomyelin 34-1. It's a well-known glomerular marker. We're really interested in it because we think that it may have a structural component in maintaining the slit diaphragm. And as we can kind of see it in this three-dimensional rendering, we see that there's heterogeneity of the sphingomyelin 34-1, not just across different glomeruli, but as you go through a single glomeruli, it seems to be distributed slightly different depending on where it is in that plane. And so we think that this type of projection, this type of rendering is going to allow us to dig in and ask more detailed questions. And then if we can bring in our orthogonal workflows, including Xenium and Codex, and do that cross-data mining and get some transcriptional information as well as some protein information, we could start understanding cell types, cell states, lipid distributions, and really start understanding true structure function of a given tissue. And so what I hope that I've conveyed to you today is that we have developed and really put through the paces our multimodal molecular imaging pipeline. In addition to the modalities shown here, we've also recently brought online 10x transcriptomic, including Visium and Xenium. And from this pipeline, just using the 2D approach, we can drive in situ biomarker discovery. And then, of course, taking it that next step into the 3D realm, we can start really getting at some fundamental biological questions. So I just wanted to thank the Vanderbilt Biomic team, and I'm happy to take any questions.
Thank you for this great talk. Melissa, are there questions?